All right, hey, welcome back. We're going to continue on with scientific method. Then we're going to talk about tools, because I don't think we'll get through the characteristics of life. So we ended last time with Pasteur's experiment. He did an actual laboratory experiment where he put broth in swan neck flasks. He heated it to kill the microbes, and he found that hey, nothing formed. Until he broke it off and let more air in, the microbes in the air could come in, and then the broth would spoil. However, lots of times in our scientific method, we can't actually do an experiment. Okay? Sometimes we can't do a controlled experiment in a lab. There's a number of reasons. Sometimes there's just too many variables to control, too many things to look at. Jane Goodall is a good example of somebody who made their life off of doing field studies, field observations. She looked at chimpanzees in the wild, studied them, and saw how they behaved. Now, observing them in nature allowed her to see actually how they interact and how they behave. Had she have taken a bunch of chimpanzees, put them in a cage or in a zoo, they would have reacted differently and we wouldn't have learned as much. Okay, so sometimes field studies are what needs to be done. Other times, there's ethical issues that preclude us from doing experiments. Say I want to show that cigarettes cause lung cancer. Can't really do a controlled experiment with that because part of that would be giving a bunch of people cigarettes and seeing if they get lung cancer. That would be unethical. So instead, what we do in those situations is we look at past data. We look at all the instances of lung cancer coming into hospitals and their past history, and we see a correlation between a lot of people with lung cancer were also smokers. So we put those in together. Also, with these experiments, with field observations, we can have large samples. Sometimes, if we were trying to do this in the lab, we would have to have too many samples to make it feasible, whereas in the wild, in the field, we can have lots of different samples. And the other time that field observations are better is when it's impossible to identify all the variables. So if we were looking at what a plant needs to grow and we brought it into the lab, maybe that would change some of the variables, like what's in the soil, okay? What's in the water source it's getting. We might not identify those variables and that would change things. So lots of times, field observations, field studies are done in place of experiments or at that step. All right, so the formal scientific method breakdown, we can simplify it into about 5 steps. You have a problem. You ask your question based on your knowledge, what you want to figure out. You form a hypothesis. It's a falsifiable question. It's something you can test. I can figure out if this is right or this is wrong. I form an experiment. Okay, that one's actually not here. That would be right here. Form an experiment to figure that out, or do a field study, or observe something. Okay? I analyze the data from that experiment or field study, and then I figure out a conclusion. Did my data reject my hypothesis or support my hypothesis? And guess what? Thing I love about this little diagram is notice that both yes and no tell me I've gained knowledge. In order for the scientific method to be good, it doesn't have to come out right. My hypothesis doesn't have to be correct. Even if it's wrong, I still learned something, and I should still communicate that with the world. Okay? That way, they don't try what I did and find out it's wrong again. Okay? So even if your hypothesis is refuted, that's still good. We still know more. Also, it kind of got broken off, but after we gain knowledge, we probably will have a new question, and so it's a big loop that just continues to go. Okay? One more thing to write down for a vocab word is a model. This is what we did yesterday. We didn't really do an experiment. We didn't really do a field observation. What we did with the mystery tube activity last week, I guess, was build a model. And we built a model to see if our model behaved like our object or how we thought it should. All right. The last thing with scientific method is a theory. Lots of people have a misunderstanding of the word theory. Okay? A theory is a hypothesis that's been tested over and over again and has been found to be correct over and over again. Okay? The actual formal definition of a theory is a well-supported explanation that unifies a broad range of observations. So theories are really broad, but they've been supported many times. Example of a theory, biogenesis. Life generates life. Okay? It's pretty basic, but it's a theory in science, biogenesis. Okay? That's a theory. Now, when you're walking down the street in common vernacular, the word theory has come to mean guess. And so that's what we're going to try and get out of our science room. A theory is not a guess. A guess is a hypothesis. Okay? A theory is an explanation that's been tested over and over again. But they are revisable. All science, as we talked about, is our best current understanding. So when new data comes, when new experiments are designed, especially when we get new tools, which we'll talk about tools in a bit, that can change our theories, but that can change anything in science if we have better understandings. All right, we're going to skip the study of life today. We're going to come back to that in the 4th lecture. So I'll roll through that. That one's a big one. And we're going to go to the tools and procedures of science because that's a lot shorter. Okay? These are what scientists use. So the first off is a measurement system. In order to communicate across the world, we have to have a standard system of measurement, and we call that SI, or the System International, the modern metric system. And the modern metric system is based on the meter. So here we have a centimeter, 100th of a meter. And here's an inch to give you representation. It's approximately 2.5 centimeters to the inch, around there. Okay. But the beauty of the metric system is it's based on factors of 10. So there's 100 centimeters in a meter. Okay? There's 10 millimeters in a centimeter. Very easy to remember. As opposed to there's 12 feet in a mile and 5,000, or 12 inches in a foot and 5,000 some feet in a mile. It's a lot easier to remember with the metric system. That's one of the benefits. But it also aids in reproducibility. One of the big things of science is being able to repeat somebody else's experiment and get the same thing. If I use different measurements, they might not be able to do that. So that's why all of science is done in the metric system. Okay? We also have to be able to analyze our data, and biological systems are very complex. Okay? If you just think of the human body, it's very complex, but when we get up to populations and communities, they're even more complex. So a lot of times nowadays we use computers to analyze our data. We can use computers to draw out molecules, to read the 6 billion bases in our DNA. We use computers to model the weather. Our weather you get on the local channel is spit out of a giant computer model. Okay? And there's lots of different examples of how we use computers, but they're arguably something that has dramatically changed science. Another thing lots of scientists use in the life sciences are microscopes. Microscopes magnify very small things, and there's light and electron microscopes. So we're going to talk about both of those, and then the difference between magnification and resolution. Okay? So a light microscope, it can magnify 100 to 1,000 times, depending on which ones you have. We call these compound light microscopes because they have 2 lenses. They actually have a lens down here, the objective, and a lens up here, the eyepiece. And you usually have to use stains and dyes to see the living organisms. Okay, so here's a picture from a light microscope, what a cell would look like. These are little organisms in pond water, okay, probably paramecium. We can also hook a light microscope up to a video or a computer so we can record and analyze what we're seeing. On the flip side, we have electron microscopes. These, instead of shooting a beam of light through it, they shoot a beam of electrons through the object, and the electrons are even smaller, so they can magnify thousands of times. Okay. There's 2 different types. Oh man, I'm approaching 30 seconds. Transmission shoots through the objects, and scanning scans the surface kind of like a record player. This is a scanning electron microscope. You can see the outside. The key thing with electron microscopes, the samples have to be dead. Okay? They can't move. They have to sit there for a long time, so they have to be dead for an electron microscope. All right, I guess we will continue on with lab techniques in the next video. Thank you.